And it takes a moment. All right, we're live.
Cool. So, shall I start then?
I think so.
Okay. It's my incredible, wonderful pleasure to introduce Ruben Coen-Cagli for this vision seminar. It's all a little uncommon to me or unused. I'm not used to this crowdcast thing, so yeah, don't judge me too harshly here. Ruben, it's great to have you here, and we scheduled your talk for about, I don't know, 5 times over the last year.
Indeed.
So Ruben is an assistant professor at Einstein College of Medicine, and before he did a PhD in Italy, I think in Napoli, in physics. I read up, studying something about visual creativity and cognitive science. I think I have to read that up. I don't know. Really interesting. And after that, he moved to Einstein College, worked with Cordelia Schwartz, and also Adam Cohen. And after that, he did another postdoc in Geneva with Alex Pouget, and great work on probabilistic inference and how particularly the visual system works. And now he's back at Einstein as a faculty, and yeah, it's my pleasure to have you here. And I think we should just start.
All right. Thanks a lot, Alan. I still have some echo, so if you could mute your mic, let's see if that works.
I'll do.
Try now. Yeah. Okay, good. Thanks a lot, Alan, for the invitation and for the kind introduction. I'm really glad to be here, and thanks everyone for being here, even though I cannot see your faces, but I know you're there. And so we were discussing earlier, if you have questions, you can write them in the chat, and Alan will read them for me. So please do ask questions anytime. You can interrupt throughout the talk. Happy to be to clarify or explain or answer. We don't need to wait until the very end. So that said, I'll tell you today about the work we do in my lab, which is focused primarily on visual processing, and in particular visual processing of natural visual inputs like this picture that I put on the screen here. We are interested mainly in 3 levels. The first one is neural coding in the visual cortex. And we do this, we look at the activity of neurons and groups of neurons responding to scenes in collaboration with experimental labs that record and perturb the activity in visual cortex of monkeys, as well as mice more recently. We are also very interested in understanding the principles underlying visual perception of complex natural scenes. And we do this with visual psychophysics in my own lab, as well as with other collaborators. And we'll be focusing recently a lot on image segmentation and grouping. But my lab is primarily a theory computation lab, and so we try. Our main goal, our primary goal, is to develop theories and computational models of visual processing at the neural and perceptual level. And I put up here this Bayes rules equation for those of you who are familiar with it, because probabilistic inference is the framework that we work with, because that is the optimal way to deal with intrinsic noise of sensory signals, as well as ambiguity of the images. I will explain those, the importance of those 2 things, noise and ambiguity. But our work in the last 4 or 5 years has been focused mostly on what this idea of probabilistic inference to deal with this complexity of the inputs, what that implies for the variability of neural activity in cortex, as well as for perceptual uncertainty. By the way, can you see my pointer, my cursor here? Or maybe I should use this one instead, make the pointer. Yeah, now I see that you can see it. Okay, good. So, so to get started, I just want to give you first some motivation for our approach to studying visual processing of natural stimuli. I'm starting here from, the classical experiments from Ruben and Wiesel. I'm sure all of you are familiar with this. They discovered in V1 of CAT that there is selectivity for orientation of the stimulus. And, experimental work ever since then has characterized the selectivity of neurons in visual cortex to different aspects of visual stimuli, of simple artificial stimuli, to a great deal. And that knowledge has been also sort of summarized and formalized in descriptive parametric models like the one I'm illustrating up here, which give us both a way of quantifying our understanding as well as some sort of conceptual building blocks. Like there's a filter, a linear filter, that describes the selectivity of neuron. There is some sort of non-linearity applied to it, and then there is some stochasticity that gives rise to spikes and spike trains. So, of course, ultimately, though, we're interested in the transformation from natural visual inputs to spiking activity of neurons. And there's been a lot of work in the context of this kind of parametric descriptive models that that show that some progress could be made, but eventually, maybe in the early 2000s, it looked like we had reached a ceiling around 50% of the explainable variance. And then, of course, after that, a few years later, deep networks came, into the field, and they've shown that we can actually predict neural activity in response to natural images a lot better with this kind of models, and we can also gain some new insights into the selectivity of neurons, and that's great. It provides a very solid backdrop for studying visual processing. However, of course, there is a number of questions, fundamental questions that remain open that are not addressed by this kind of black box parametric approach. And so our approach is kind of complementary and distinct from the parametric approach. We term it normative approach to studying visual processing, which starts from hypothesizing some computational goals of the visual system, and from there deriving predictions about neural activity or about perception. One classical example of this is the work of Bruno Sausen and Field with sparse coding and related work with ICA, where just by hypothesizing that the goal of the V1 neurons was to implement a sparse code, an efficient representation optimized to the statistics of natural images. By hypothesizing that, they showed that you can derive receptive fields that are very similar to the selectivity of neurons in V1. So you can explain V1 selectivity by understanding some properties of images and assuming that the goal of the neurons is to represent images in this kind of efficient way. So that's very much our work is in the same spirit. But we are really interested in going beyond single neuron selectivity. We're interested in the structure of neural activity, which includes things like the dynamics of the activity of individual neurons, the variability across repetitions of the same inputs, and how that variability is shared between pairs and larger groups of neurons. And we have been hypothesizing that the structure of neural activity, this rich statistical structure, actually should reflect structure of the natural inputs themselves, the complex spatial and temporal dependencies that are present in natural images. And so to be a little bit more specific, our work, the work that I will tell you about today, starts from the idea that the goal of cortical populations is to achieve an optimal or near optimal representation, and specifically that requires two-step, a probabilistic inference. So we want neurons to represent probability distributions of the features that they encode, and we further assume that they do so by using neural sampling. That is, the activity of neurons represents, at each point in time, represents a sample from the target distribution that the neurons are encoding. And so our approach to, our hypothesis is that the structure of activity in visual cortex approximate a probabilistic representation optimized to the statistics of visual inputs. And the approach will be to, to develop detailed generative models of natural images, which are necessary to perform then probabilistic inference, to use neural sampling theory to relate inferences to neural activity, and to test the predictions with V1 population recordings. So I'll tell you today in the first part about a work that we've done to try to unify two distinct phenomena, contextual effects and response variability in cortex. I will then go on to tell you about how we can go beyond contextual modulation and study sensitivity of V1 to segmentation cues. And then if there's time at the end, I will tell you about some initial methods development that we've done to study perceptual grouping and segmentation. So this first part is work that it's based on work that I started when I was a postdoc with Odilia and Peter Dayan, and that then we have built upon that work in my own lab, in particular with my postdoc Dylan Festa and others in the lab. And I want to start by just reminding you of these two cortical phenomena that we're interested in, if you're not familiar with them. One is contextual modulation, which is illustrated here with one particular kind of contextual modulation. This is surround suppression. So in this image, the blue circle is the receptive field of a V1 neuron. And in these experiments in the Marshall lab, they present stimuli with a progressively larger and larger size, and they find that the response of the neuron, the firing rate, goes up until the stimulus fills the receptive field, and then it's suppressed by larger stimuli. So this suppression of responsivity by larger stimuli is called surround suppression, and it's one example of contextual modulation. It has been studied experimentally to a great level of detail, including its tuning, its dependency of certain features of the stimuli, its dynamics, and so on. But more importantly, surround suppression is an example of contextual modulation, but similar kinds of contextual modulation appear all throughout the visual system, both in cortical and subcortical areas, as well as in other sensory modalities. It's also been observed in the integration across different sensory modalities and in value-based decision-making beyond sensory processing. The second phenomenon that we've been very interested in is response variability. This is another sort of fundamental feature of neural activity in cortex, and it's illustrated here for one particular neuron, where we plot the variance of the activity of the neuron to repeated presentation of the same stimulus as a function of the mean spike count. And what you see is that there is a strong correlation between the mean and the variance. There is a structured response variability. It's not just noise. This structured variability is also widespread throughout many different parts of the visual system. And so our goal really has been to provide a normative account of V1 contextual effects and response variability, to try and understand if there is some sort of common origin, if they serve a common purpose. And our hypothesis is that they are both signatures of a probabilistic code optimized to the statistics of natural images. So this is what I'm going to tell you in the next 5 or 10 minutes. And to do that, I need to start from telling you something about the statistics of natural images. So we're interested in the statistics of images at the level of V1 neurons. So you could think of you could start with a visual filters, an oriented visual filter, similar to the input to a V1 neuron, and apply this filter to many patches of natural images. And what you would find consistently is that the histogram of activity of the filter is very sparse. So most of the time the filter is silent, but occasionally it will give a very strong positive or negative response when there happens to be a feature in the image that matches the filter. Now, one property of natural images is that, and that's something that was studied by Eero Simoncelli's lab 20 years ago now, is that if you're looking at high contrast regions of images, you'll find much broader response distribution as opposed to low contrast images. And that in addition, if you're looking now at two filters, one, let's say, representing one location corresponding to the center, to the receptive field of a neuron, and another one corresponding to a location and a feature in the surround, then these two observations are statistically dependent, which is summarized by this bow tie shaped conditional histogram. And what it tells you essentially is that you can, to some extent, predict the variance of the activity of one filter by just looking at the magnitude of the other filter. If one filter has a magnitude close to zero, chances are. Sorry?
Can you hear me?
Yeah, I can hear.
There's a question. Oh, sorry. There's a question from Johannes
Yes.
Birch. Here, the distribution of responses is over natural image patches, not repeated presentation of the same patch, correct?
Yes, that is correct. And sorry, Alan, if you can mute your mic because I have a strong echo. Thanks. So yes, that's correct. We're looking at the statistics of the output of filters applied to different patches of natural images. In a couple of slides, we will get to how that relates to variability of neural activity across repeated presentations of the same patch. But for this slide, we're looking at statistics across different images. And it was found that there is this strong spatial dependency between two filters. So also other work by Labovero, Simoncelli showed that there is a very simple generative model that can explain this statistical dependency. That is, if you start from a pair of independent local features, GK, GS, and you multiply both of them by this shared random variable, which you could think of as representing the contrast level of the patch. Then across many patches, you would vary this value of V randomly up and down, and that would produce this kind of spatial dependency between K and S. So there is a simple generative model which has this multiplicative structure that takes us from independent local features to dependent observations. And the reason that that's important is that, as I mentioned earlier, our hypothesis is that the goal of neurons is to represent the probability distribution of the G features of the local features given the observed inputs, and that to compute this quantity, this target distribution, we need to know something about the process that generates the observations in the first place, so that we are then able to invert it. So in the next slide, I'm going to focus on a couple of properties of this posterior distribution, which explain some aspects of contextual modulation and response variability. And so just to make things a little bit more concrete, when we actually implement these kind of models, we have a filter that represents the center of the receptive field of the neuron, and we have a group of filters that represent the surround, with tuning that and location and size that are based on empirical observations from V1 physiology. So again, the goal of the neuron is to represent this target distribution. And in the neural sampling, under the neural sampling hypothesis, what that means is that the moments of this distribution should tell us something about the statistics of the responses of the neuron. And so, in particular, the mean of this distribution should be predictive of the firing rate of the neuron. And the variance of this distribution should be predictive of the variability of the activity of the neuron across repeated presentation of the same input. So in past work, we derived analytical results. And by the way, I should say this slide is going to be a bit dense and technical. Apologies for that, but I think there are a couple of insights to gain that are really worth it. And then we'll go to more experimental data. So the first analytical result that we had a few years ago was that the mean of this posterior distribution is approximately given by a ratio between the feed-forward input in the receptive field and this quantity, which is a combination of the energy in the center and in the surround, and turns out to be an estimate of the global contrast level. This, of course, this explains surround suppression, because when you increase the size of the stimulus, after you exceed the size of the receptive field, the only thing that changes is the surround energy, and that which increases the denominator and therefore decreases the mean response of the neuron. And we showed in past work that once you implement this model in detail, you can also capture a great deal of tuning properties of surround suppression in actual data. In more recent work, we started looking also at other aspects of the distribution, and in particular, we looked at the variance of this posterior distribution. And it turns out that, again, analytically, we can show that it is also, it has a very similar expression to the mean, except with different exponents. And it's also, it has also this kind of divisive form. So first of all, it captures inference in this GSM model captures the relationship between the mean and the variance of the response across different stimuli. It explains why they are related to each other. And it also provides a sort of unifying link between the contextual modulation of firing rate and of variability by way of divisive normalization, suggesting that maybe there could be a shared mechanism that controls contextual modulation and response variability. I should mention that these results cannot be captured by an additive model if we assume that there was an addition here rather than a multiplication. So it seems that the fact that inference is based on the generative model that capture the statistics of images is really necessary to unify contextual modulation and response variability. And then one more important thing to notice is that the surround stimuli, which intuitively reduce uncertainty, right? Sorry, there's a question. Oh, I see.
Yeah, Ruben, sorry. There's a question from Lingchi Chang. What are the assumptions of filter S, K, and S? Of filters K and S? Are they of random orientation, frequency, band, phase, etc.?
Thanks. So, sorry, Alan, again, if you could mute your mic. All right. Yeah. So the center filter here is just, let's say, vertical with a given spatial frequency, and it's just one filter. And in this particular set of simulations, I'm assuming that surround filters all have the same orientation and spatial frequency. In past work, some of this published here, we worked with different configurations of the surround, for example, where we have multiple orientations in the surround. And then when we apply these kind of models to actual neural data, we have to, for example, look at a center filter that matches the selectivity of the individual neuron. I'll get there in a couple of slides. But at this level, where we're just trying to reproduce this qualitatively, these properties, we simply assume that the surround is roughly matched to the center in terms of frequency and orientation, which captures to a first approximation what we know empirically from studies of surround suppression. Although, again, we know that there's a lot more complexity to it, and we have captured some of that in these older publications. So I hope that answered the question. So they're not random overall. I see that there's another question from Yannis, so I'll just read it aloud. The expected value of G is the expected response of the filter to the image conditional on the observation of K and S, assuming a contrast normalized image. So the first part, yes, the expected value of G is the mean of the posterior distribution of G given K and S. we don't assume a contrast normalized image. It's just an unprocessed image. And the the normalization happens here. Although you can do the same on you can apply some pre-processing to the images and do a very local contrast normalization, and then apply this GSM framework, and the GSM still captures well the statistics of those inputs. All right, so I emphasized here the role of surround stimuli. I was saying that intuitively, when you present a larger stimulus, you're providing more information about the target feature, and that reduces uncertainty. That's intuitive, but also we have shown that analytically. But the prediction that follows from that is that in the neural sampling hypothesis is that the variability of activity should be suppressed by surround stimulation. And so I'm going to move now to some experimental results where we tested actually this prediction. These are data collected in the lab of Adam Cohn by some of his students, where we present images that are either small, about the size of the average receptive field in V1 at the eccentricity where we record, and then also larger, much larger, so that they extend well beyond the suppressive surround. So this is one example neuron, and each point is the response to an image, either small images or large images. And what we see for this particular neuron is that on average, large images suppress the spike count, suppress the average response, as we know from a lot of past work. And then we also quantified the variability using the Fano factor. That's the ratio between the variance and the mean, and it quantifies changes in variability beyond those that could be explained by simple change in the mean activity. And what you see for this particular neuron is that across large images, on average, the variability is smaller. The Fano factor is smaller than it is for the smaller images. So we can now build a scatter plot of Fano factors for large and small images, and what you observe is that across all the neurons that we recorded from, the vast majority had much lower variability, much lower Fano factors when they were stimulated by large images compared to small images, supporting our hypothesis that surround stimulation should reduce uncertainty, and in a sampling-based code should also reduce the variability of activity of neurons. So this was a sort of an initial support for the hypothesis, but of course there are many caveats and many controls that one needs to run. I will not go through all of those, but at least one is very important, and it's based on the fact that we record with an array, and many neurons are not perfectly centered on the stimulus. So if you think about it, when you present a larger stimulus, some of the image will actually go inside the receptive field of the off-centered neurons. And we know from past work, that famous paper by Mark Churchland and many other authors, that stimulation of the receptive field itself stabilizes the responses of V1 neurons. So we wanted to know if the effect that we're seeing is only due to stabilization inside the receptive field, or there's actually a contribution outside the receptive field. And to test for that, we measured the size tuning by increasing progressively the size of an image. And we observed for the firing rate the typical non-monotonic behavior. The response goes up, and then larger stimuli suppress the firing rate. But we observed that the Fano factor was monotonically reduced by increasing the size of the stimulus. So there is a drop in variability due to stimulating the receptive field, but there is also an additional drop of variability that is due exclusively to stimulating the surround of the neuron. And we quantified this both with natural images and gratings, and both in awake and anesthetized animals, to control for a number of other possibly confounding factors. So overall, we can confirm that surround stimulation reduces response variability more than it reduces just response strength and beyond the stimulation of the receptive field. So to wrap up this part, I've tried to show to you that if one starts from this hypothesis of that neurons in V1 aim to represent a posterior distribution of features that they encode using a sampling-based representation. And if this is based on a model similar to the GSM, which captures the statistics of images using multiplicative interactions, then you can explain contextual modulation, surround suppression, and you can also explain the dependency between mean and variance. So you can explain structured variability for individual neurons. And you have this sort of fundamental link between the two of them through divisive normalization, which suggests that there may be shared mechanisms for the modulation of variability and the modulation of firing rate by context. And lastly, predicts that surround stimuli suppress variability, which we have validated with experimental data. So unless there are other questions on this first part, I'm going to move to the next one. Okay, I don't see questions in the chat, so I'll move on. So the second part is work that, again, I started a while ago when I was working with Adelia and Peter, and that we're building on that work now to understand something about variability, covariability, and dynamics of neural activity in V1. And the initial observation, the initial consideration was that the GSM, yes, is an effective model. It captures statistics of images, but it's an extremely simple model. We know that images have much more complex structure than just these multiplicative interactions. For example, one key aspect of images is that they're non-stationary. So for example, when you're looking at the center and surround filters that are looking at the same object, same physical object in the scene, then the GSM could be a reasonable model of the statistical dependency. But when the center and surround filter are looking at different physical objects or an object and the background, then you would expect that they are much more statistically independent, and they would be better captured by a model with independent mixer variables. So we call these kind of images spatially heterogeneous and these kind of images spatially homogeneous. And our prediction back then was that the surround modulation should be only activated or more strongly activated by homogeneous than by heterogeneous images. And so we tested this, and this is work that we published a few years ago now, again with recordings in Macaque V1, where we presented many different images, and for each one of these images, and for each turn, we determined whether the image was heterogeneous or homogeneous based on its statistics. And here's where it's really important that we empirically measure the receptive field properties of the neuron, because the filters that we use for the inference will determine whether an image is homogeneous or heterogeneous in some cases. So that answers one of the earlier question by Lin-Chi-Zhang, I think, yeah. So we did this. One thing that's important to notice is that this model that the M-GSM, the mixture of GSM model we use to infer whether an image is heterogeneous or homogeneous, it's not trained on neural data. It's not trained on the images that we present in the experiments. It's trained on a separate database of natural images. And so this labeling the images as heterogeneous or homogeneous is actually a parameter-free prediction of the strength of surround suppression. So we found that there was strong evidence for this prediction. This is one example neuron where we quantified the strength of surround suppression by the modulation ratio, and we showed that on average across heterogeneous images it was much weaker than across homogeneous images, and this was true across the population of neurons that were recorded from. In addition, we could take this insight and use it to extend descriptive models, and at the time we could achieve state-of-the-art predictive power based on this insight about statistics of the images. So therefore, now that we also know from our, the first part of the work that surround stimuli also modulate response variability in the way predicted by neural sampling, we reasoned that we should have a similar sensitivity to this kind of homogeneity or to this kind of segmentation cue in the variability of neural activity. And that's because in this framework, surround stimuli reduce uncertainty only when the stimulus is homogeneous, not when it's heterogeneous. And therefore, they should only reduce the variability of neural activity in that case. And sure enough, we're finding strong evidence for this. In here, I'm showing the Fano factor for large heterogeneous images versus large homogeneous images. And you see that both in awake and anesthetized animals, we find that Fano factors are consistently larger, so there is higher variability of the responses of neurons when images are heterogeneous. I should mention that each point here is the summary, is the average of a population of neurons. So there's usually between 10 and 40 neurons in each one of these points on the scatter plot. So that's interesting, but one observation that we found even more interesting and maybe to some extent counterintuitive is that this inference of homogeneity can shape the response, the structure of the responses of individual neurons even beyond the variance, the mean and variance. And so the thinking was, well, if you have homogeneous image, it's homogeneous. A heterogeneous image, it's heterogeneous. But actually, these properties of homogeneity is itself based on a probabilistic inference. You only observe the filter activities, and you have to infer whether they're homogeneous or heterogeneous. And that means that there have to be somewhere in between some images that we term ambiguous, some images where the probability that the surround and the center are homogeneous is somewhere around 0.5. And the reason that these ambiguous images are interesting is that they predict for V1 neural activity something similar to what we observe perceptually with perceptually ambiguous images. Like the image, if you keep staring at this image on the screen, sometimes you will perceive a duck, here's the beak, and sometimes you will perceive a rabbit, here's the mouth of the rabbit. So if you keep staring at it, your perception will shift. It's a bistable percept. And our idea is that when an image is ambiguous with respect to surround homogeneity, even though it's not perceptually ambiguous, it should induce a similar kind of bistability in the responses of V1 neurons. And so we tested this, and to test this, we used similar experiments, but where we now use a much larger number of repetitions of each image so that we can accurately measure the response histogram. This is the response histogram here for one example neuron responding to one of our ambiguous images. And you can start to see pretty clearly the presence of two peaks, which is, intuitively is consistent with our prediction of bimodality. So, of course, we want to quantify this, and we do this by model comparison using a framework that we have developed recently to work with discrete mixture of Poisson and Comp-Poisson models. And so we have two models that are essentially describing unimodal kind of standard response distributions. And then we have two models that capture binary mixtures that can capture this bimodality. For the example neuron here, you can see that the best model with cross-validation is model number 4. It captures very well the shape of the response histogram. And you can also see that the two individual components, the two mixture components with the dashed lines, capture very well the two peaks of the response histogram. Whereas for the same neuron responding to a known ambiguous image, you can see that the response histogram is clearly unimodal, and it's best described by either Poisson or Comp-Poisson distribution. In this case, they are essentially equivalent. So, of course, we wanted to quantify this across neurons and images, and here are the results summarized in this graph. And there are two things that I think are interesting to observe. The first one is that the vast majority of cases, the vast majority of neurons and images are best captured by the standard Poisson or Comp-Poisson models. So they're unimodal, which, there's a number of reasons why that might be the case, but it's also reassuring because certainly bimodal distributions have not been observed that often in V1. The other observation, though, is that when you have evidence for bimodality or for mixtures, then that's twice as likely for ambiguous images than it is for non-ambiguous images, which provides some support, some evidence in favor of our hypothesis that these mixture responses, this kind of bimodality is a reflection of ambiguity, intrinsic ambiguity of the images themselves. And so one thing that we are working on right now that's I think very exciting and very interesting, and we only have some preliminary results, is the dynamics of the switching between the two different components. So when you have a histogram like that, is it that the neuron is sampling, around one mode for quite some times, and then it jumps and it goes around the other mode and it keeps jumping around here, or is it rather than the neuron's activity jumps back and forth between the two modes quickly? That's interesting because, depending on the answer, you could then think of very, very different mechanisms that could implement this kind of bistable activity in V1. And what we're finding so far is that the states transitions can be very fast, on the order of a few tenths of milliseconds. Again, I will not explain the details of how we do this, but if you're interested, we can talk about it more later on. So to wrap up this part, the first two parts, I have proposed to use a normative approach to relate natural image statistics to the response structure in V1, to go beyond what we know about just the selectivity of individual neurons. I've shown to you that this theory of neural sampling based on inference on a GSM can unify widespread cortical phenomena of contextual modulation and structured variability. I've shown to you that the link between the two really is divisive normalization. And given how widespread divisive normalization is as a description of contextual phenomena, this could be a much, much more general result than just being V1 specific. And it also, because it suggests that normalization plays a key role in probabilistic inference. I've also shown to you that V1 is sensitive to segmentation cues, and that this sensitivity goes beyond just the firing rate of individual neurons. It's true for the variability, and it's also true for higher order properties of the response statistics, including some implications for the dynamics of V1 activity. So I don't know if I have another 5 or 6 minutes. Alan?
I think so, definitely. Sorry for the echo.
All right. Thanks a lot. Great. So in the last 5 minutes, then I'll just briefly tell you about how we're planning to study perceptual grouping and segmentation of natural images, and this is work that. For which I should credit the French mafia in my lab and around. So Jonathan and Claire, Jonathan was a postdoc with me, and Claire just started recently, and this is work done in collaboration with Pascal Mamassian. So our motivation for this project is that there is, perceptual segmentation and grouping is a fundamental kind of task of the visual system. That has been studied extensively with simple stimuli, and we have, a number of principles have been discovered for perceptual segmentation, like Gestalt principles of proximity and similarity. There's a lot of work that suggests that these principles also to some extent reflects the statistics of natural images. There's evidence that different cues for grouping or segmentation can be combined in a near optimal fashion, and of course there are also top-down influences on our perceptual grouping and segmentation. So there is a lot of knowledge that we have, but we really are interested in perceptual segmentation of natural images. And we realized a couple of years ago that in order to make progress in that direction, we really need to address two shortcomings of the existing literature. One is that we lack ideal observers. for natural image segmentation. So we do have segmentation algorithms in the computer vision community, but not much time to what we know about cortical processing and perception. And on the other hand, we also lack controlled measurement of perceptual segmentation maps. All the work, all the databases for segmentation that are out there are based on manual tracing of natural images. So I show you an image on a tablet, and then you use your hand and a pen to actually draw on it. And that introduces a number of problems and limitations. And so in the next couple of minutes, I just want to tell you about the work that we've done to develop methods to address these two shortcomings. And we're right now in the process of applying these methods to study segmentation in humans. So the first part is that we have developed probabilistic segmentation algorithms, but with the goal of combining, some of those perceptual principles based on the literature with simple stimuli, with knowledge of image statistics and knowledge of cortical processing of image statistics. So by now you know about the GSM and you know about the mixture of GSM. And our idea was that that could provide a starting point for a segmentation algorithm that satisfies these two properties, right? Because the GSM is a good model of local image statistics, and the M-GSM can capture how those statistics vary across segments or objects in an image. So we started from that, and to introduce some of those Gestalt principles, we use a formulation in which we have a prior distribution on the segment label of each pixel. And we formulated this model in such a way that we can now combine information about the segment of one pixel with the information about segments of another pixel in the prior. And we can do this, and the advantage of this is essentially that we can do it in a way that's computationally efficient. It's a linear update rule, and that it's based on uncertainty. So essentially what we have implemented here is some sort of spatial smoothing of segmentation maps, but it's a spatial smoothing that's based on where the information from neighboring pixels is weighted by the uncertainty about segmentation at each one of those pixels. And so we first implemented this model using V1-like features as our observations in this model, and we could obtain decent results for some images. But of course, for some other images, we just cannot capture the complexity of the features based on which we segment images. And so we extended this to a hierarchical version of the algorithm, where the features now are not just V1-like oriented filters, but they're actually taken from different layers of a pre-trained deep network, which we know captures increasingly more complex features that are also relevant for human vision. And of course, the advantage of our framework is that we formulate this as a probabilistic inference, and we have this uncertainty-based combination rules illustrated here by the red arrows, where we can now combine information about segmentation not just across different neighboring pixels, but also across different levels of the visual hierarchy, including both in the feed-forward and in the feedback direction. So we have, in order for that to be viable, first of all, we had to show that deep convolutional neural network features were actually well described by GSM models, and we've done that in a paper that came out in NeurosCience a few months ago. And then we can implement this, and here's one example segmentation map at the different layers of the deep net. And one very interesting property of this algorithm is that you obtain segmentation maps that are consistent across the visual hierarchy in such a way that at the bottom layers you are using some information about higher level features to obtain a more accurate segmentation. And at the deeper layer, you're receiving some higher resolution information from the lower layers, so that you can, capture these high-level features without losing too much resolution. And I think this algorithm now can approach state-of-the-art performance on the BSD segmentation database. But it's also, I think, a very rich. It provides a very rich data set to study feedback and feed-forward interactions based on uncertainty-based feed-forward and feedback interactions for segmentation. So we're planning to build on this to generate predictions about segmentation-related phenomena in cortex and in visual perception. And lastly, just to tell you that we have also developed a new experimental protocol to measure perceptual segmentation maps. Following a well-controlled visual psychophysics standards. And the way it works is that we ask subjects to divide an image in a prefixed number of segments, or we can also do that without specifying the number of segments. But then we show an image, and then we show two dots, and we ask subjects to tell us whether the two dots belong to the same segment or a different segment. And we repeat this a number of times. And then by solving a system of quadratic equations numerically, we can, if we have enough measurements, enough pairs, we can actually reconstruct the perceptual segmentation map of the individual subjects. And here's one example for this image for one subject. And we can also reconstruct the probabilistic segmentation maps underlying this one, right? This is the probability map that each pixel belongs to segment 1 or segment 2 or segment 3, which is important because, again, this gives us a way of measuring measuring perceptual uncertainty and comparing that to uncertainty in our model of segmentation. So, so I'm going to stop here, and I want to again acknowledge all the people who have worked on this. In particular, my postdocs Dylan Festa and Sasha Sokolowski and Jonathan Vacher, and my collaborators and funding sources. And thanks everyone for the attention. I'm happy to take questions. So I see there is a question. Should I just read it aloud?
Thank you, Ruben, first of all. And yes, I think it's best if you read the questions, and then we don't have this awkward back and forth. Read the questions, please.
All right. Thanks. This is from Lingxi Zhang. Thanks for the talk. Thank you. Could you provide some intuition behind the relationship between mean firing rate and variability in your framework? Does a higher feature activation actually imply more uncertainty? And then related to that, do these properties depend on the image being naturalistic? Could you in principle design adversarial stimuli that drive the model to a different regime? So great questions. Thanks a lot. I'll start from the first one, which is what's the intuition behind the relationship between firing rate and variability in the GSM framework, and whether that implies that a higher feature activation also implies more uncertainty. And so I have a slide where I can actually illustrate that. So, which is this one here. So here, here I'm illustrating with a black line the joint posterior distribution of the local feature G and the global mixer variable V given a stimulus. And because remember that in the model, the observation, the stimulus, the observation X is G times V, once you observe X, G and V can only take values that are actually on this line, right? Their product has to be constant, and so they can only take values on this line. Now, if you have some, for a given stimulus, you have some guess about what the value of the mixer is, and let's say that it's this, this yellow posterior distribution for the mixer, and you have some evidence that the mixer has a low value with some uncertainty, then the corresponding distribution of G, once you marginalize out the mixer, is just the projection of the corresponding chunk of the joint distribution onto the G axis. And you get this broad yellow distribution for G. Now, if you have evidence for a larger value of the mixer V, for example, by providing a larger stimulus, so there's stimulation now in the surround, the distribution for the mixer now shifts to higher values, and the corresponding chunk of the joint distribution, when projected onto G, correspond to a much smaller range of possibilities. Right. So in other words, if you know that the mixer value is small, then there is a large range of possible values for G. But if you know that the mixer value is around some larger value, then there is a much narrower range of possibilities for G. And this is what couples the mean and the variance of the posterior distribution for the feature, right? However, and this is illustrated here, there are different ways in which you can change the stimulus and change the mean and the variance. So for example, you could change the contrast of the image, and that would change both the numerator and the denominator, both the center and the surround inputs. And that will lead to an increase of the mean with a slight increase of the variance. You could have a change in the orientation, where you would only scale the strength of the center input but not the surround, which will lead to a large increase of the mean and the variance. But you can also scale just the context, the surround, in which case you would only scale the denominator, and that would predict a reduction of mean and variability. And the other thing that I should mention is that all of these intuitions are based on an analytical version of the GSM, where we assume that there is no observation noise. In other words, that the observations X are exactly the product of G times V. But a better model is one where there is also observation noise. So the X is G times V plus some noise. And that is also better matched to neural variance to mean relationship, because in this case, when the input goes to 0, the variance does not go to 0. It's actually bound by the observation noise. So, so I hope that clarifies a little bit the intuition for why in this framework the multiplication in the generative model implies a relationship between the mean and the variance in the posterior. And so going to the second part of the question, do these properties depend on the image being naturalistic? So they do not depend on the input image being natural. So these predictions also apply for gradings, and we've tested that also empirically. What they do depend on, though, is they depend on the GSM. They depend on the fact that the inference is based on a model that captures natural image statistics accurately. And so, if, for example, if we have if we have an additive model where the V and the G variables are summed together rather than multiplied, you would not be able to capture the fact that the variance depends on the stimulus. The variance would be stimulus independent. So this is a very important distinction. The input for which you compute the inferences can be anything, naturalistic or artificial, but the inferences need to be tuned to the statistics of natural images, which in our world means that they have to be based on something like a GSM. They have to be based on the fact that there is this multiplicative latent interaction. And then the last part of the question is, could you design adversarial stimuli that drive the model to a different regime? I think it's a great question. I have not thought about it. So I don't have a good answer for that, but it seems like something very fun to think about. So I will definitely look into that. Thanks for the suggestion.
Okay. Are there any more questions? I think we still have a couple minutes. I have a question. I just, rather than typing it, I just ask it directly. So you were saying basically that the spying rates are sampling to the posterior about the image feature, right?
Right.
Now, how does contrast play a role in that whole thing? Because contrast doesn't really change the uncertainty about the feature, right? Or,
Well,
I mean,
yeah,
yeah, go ahead.
Okay. All right, now I was just going to say that, yes, the contrast, increasing contrast intuitively should reduce uncertainty about the features.
It should not, right? I mean, the question is the inference about the feature out in the world there, or is it about some earlier signal in the neural pathway? Because I think contrast is only affecting the uncertainty once we think about the signal in the neural pathway, not about the stimulant there. Or I don't know. That's the way we usually think, right? We usually think of contrast as being a modulator for internal noise, so to speak, right?
Yeah.
So I'm not quite sure whether I understand how you think about that in your probabilistic framework.
Yeah. No, I understand now. And sorry, can you mute your mic again?
Yeah.
Sorry about that, but it's very hard to talk with the echo. So no, you're right. I forgot to mention that yes, the reason that in this generative model, the reason that contrast decreases uncertainty is that we actually assume that there is input noise. So the full generative model, I didn't talk about the full generative model much in the talk because for surround effects, it doesn't really matter. But for contrast effect, it's key that you also correctly account for the fact that there is sensory noise. And so you have to have some sensory noise in the generative model. So generative model is G.V plus noise. And so when you have very low contrast, you only have noise. And as you increase contrast, you have lower uncertainty about the features. So that's correct. And so in a way that, yeah, that reflects something about the feed-forward inputs.
Okay.
I think there is another couple of questions, so I'll read them. The first one is from some anonymous. Given the dependence between nu and g, how do you go about measuring these statistics from the image set? So I'm not sure measuring which statistics, but so there are, let me try. So there are conceptually two steps, right? One is training the model, one is learning, and the other one is inference once the model has been trained. So for training the model, what we do is we have a large collection of image patches, and we are optimizing the parameters of the model, which are the covariance matrix between the Gaussian variables. So that's the prior for the G is a Gaussian, correlated Gaussian, as well as the parameter of the distribution of the nu variable, of the V variable, which is in our case a Rayleigh distribution, or sometimes we also use a gamma. It doesn't make much of a difference qualitatively. So you can compute the likelihood of images of our filter outputs given under the GSM given the value of those parameters, and then you optimize the parameters by expectation maximization. Or when it's a simple GSM, you can do moment matching. When it's a mixture GSM, you need expectation maximization. But in any case, what you do is you just look for the parameters of the prior that maximize the likelihood of the images. Whereas when you're doing the inference on a single input, then in the noiseless GSM, that can be done analytically. So we just compute the posterior distribution analytically by solving the integrals for marginalizing V or nu. For the model that includes noise, we have developed a couple of ways. One is semi-analytical, the other is sampling-based. So we draw samples from the posterior using probabilistic programming, and we draw many samples so that we have an accurate representation of that posterior distribution. I don't know if that answers the question. I might have misinterpreted the question, so if so, please, follow up and I'll try to answer better. But now I'm going to switch to the next question meanwhile. The theory predicts firing rate and final factor data well. What about known characteristics of pairwise spike count correlations from Takahiro Doi? Thanks a lot. That's a great question, and that's definitely something that we're very interested in. We've been working on it for a while, and we have some preliminary data. I have a new postdoc that just joined the lab that is devoting his efforts to that. We have some evidence that there are changes in noise correlations that are consistent with the model. I can show you just a couple of examples. But one thing that I should mention is that whereas the predictions for single neuron are in a way straightforward, the predictions for pairs of neurons are a bit trickier because they do not depend only on the image and the tuning of the neuron. They also depend on the relationship between the two neurons, whether the two neurons have similar or different tuning. And I want to illustrate that with this slide here, right, where we find that if we're looking at two neurons with overlapping receptive fields and with similar orientation and spatial frequency tuning properties, then large images tend to reduce the correlation coefficient. Whereas if we're looking now at two neurons with receptive fields that are spatially separated, stimulating the surround actually tends to increase the correlation coefficient, the noise correlation coefficient between the two neurons. And I think this is fits very well intuitively with the sampling-based framework, because in this case, when you have a small image, the center and the surround neurons are looking at two physically different objects and statistically independent. So the posterior distribution of the two features is uncorrelated or nearly uncorrelated. Whereas when they're looking at two parts of an image, sometimes, on average, sometimes they will be looking at the same, homogeneous input, and for which the posterior distribution will be more highly correlated. So in a way, you can think of this as another way of looking at the sensitivity of V1 activity to segmentation cues, but now it's on a pairwise level. And of course, you can elaborate and extend this to populations of neurons and the intrinsic dimensionality of their activity. So definitely, there are predictions for noise correlations. We're looking into that, and it's a very exciting direction to go into. I see another comment or question from Johannes. So following up on Lingxi's and Alan's questions, I'm trying to get a better intuition for why you want to recover G and what you learn once you've recovered it. Is the aim to recover, given input noise, the probability of the presence of a feature in the image that bears a certain similarity to the filter's preferred feature? So so this is I think this, it's a good point emphasize that we're thinking about a sort of a general purpose representation of the image, as opposed to one that is kind of informed by a task. And now, Yannis, I'm referring to your recent review paper, which suggests correctly, and I totally agree with it, that we should really think about tasks in order to understand processing of natural inputs. But in this case, we're looking at early visual cortex, and we're looking at what we can learn about it by just thinking about the general purpose representation, rather than one that is optimized for any specific task. Although then in the segmentation work, we're really looking into how the task can actually then influence and modify the activity in the earlier layers. But anyway, so here the goal is to recover the intensity, how strongly a specific feature contributes to the image, to the input image, and that that is a representation of the strength of the different features, of the different orientations and frequencies that compose the input image, and then based on that inference, you could build a different kinds of readout to solve different tasks. I hope that answers the question. Mostly yes. Perhaps I will follow up. All right, let's do that.
Last chance. Okay, if not, I'm speaking for probably everybody that we really enjoyed the talk. Thank you a lot, Ruben, and thank everybody to be here, attending, and yeah, I don't know, speaking to a box here, it feels so real. And, see you next talk.
Yes, thanks everyone. Thanks again for the invitation, and thanks for the great questions and feedback. And I hope to see you all in person at some point, maybe at conferences or visits when the world reopens. Bye, everyone.